welcome to the channel. So in today's video, we are going to learn how to select a novel plant for phytochemical research. It's a big questions every B.Pharm, M.Pharm and the PhD students having when they're starting their research. So choosing the right plant is one of the most important steps in any phytochemical study. So the poor selection can lead to repetitive works, while the right approach can help you identify a truly novel research topic with a good publication potential. So in this video, I will guide you through a simple step-by-step process from finding plants available in your region, checking their traditional uses, exploring phytochemical and pharmacological studies, identifying research gaps, and even selecting a plant that has genuine novelty. And I will be also discussing few of the databases that can help you in your research much easier. So if you're planning to start a phytochemical research project, this video is for you. So let's get started. So why do many research projects fail? So the most of the reasons are like research project fail not because the experiments are poorly conducted. It's because the research question is not novel or well planned. So researchers often choose a plan that has already been extensively studied, fail to identify a real research gap, or start without a thorough literature review. So sometimes the selected plan is not readily available, lacks proper authentication, or doesn't have enough scientific justification. So, so the finally the study becomes repetitive, difficult to publish, or fails to make meaningful findings. So that is why selecting the right plan is the foundation of successful phytochemical research. The reasons I have also mentioned here: selecting a common plant, or no literature review, repeating already published works, no clear research gap, poor experimental planning, plan unavoidable or difficult to authenticate, and weak scientific rationale. So, these are the few reasons your research gets failed. So, the first step is to find plants that are available in your region. There is no point choosing a plant that you can't easily collect. So, start by making a list of medicinal plants that grow in your state or nearby areas. You can get this information from local flora books, botanical gardens, state forest departments, botanical gardens, the universities, tribal knowledge, or the flora books. Even some online databases are available, like a GBIF. I think you heard about it. It is actually about the global biodiversity information facility. So from that databases you can collect the data. So once you have a list of locally available plants, you can move on to the next steps, just checking what research has already been done on each plant. So that is a main thing. So you need to remember one thing: availability will comes first. So a locally available plant makes your research easier. So it is actually a more practical and cost effective. For example, you can see here in the Gujarat, like acacia, neem, and the calotropis, there are many drugs are there. So these are like available in that particular region. So first of all, you need to find out that which plant is, what are the plants actually available in your locality. So you can check in this all the sources. So if you find the plants from there, collect all the plants name, then you can go for the second step. So the next step is verify plant identity. So identification of the plant is very important in your research. So why the plant identification is very important? So it's because many plants may look similar, and by using the wrong plant affect your end research. So it is very important part. So the first step is a proper collection of the plant specimen. So you can collect the parts from the location, and you need to know the date of collection and the habitat of the plant. So the scientific name you will get, that you can get from anywhere, and the family of it, local name. So vernacular name, maybe you mostly heard about vernacular names. So locally it has many different names, And authentication is also the next very important. Authentication means once you have collected the plant part, we need to confirm that whether the plant you have collected it's the proper one, the correct one or not. So for that you can go and visit the botanist or the taxonomist. So with the herbarium, they will study the morphological structures like the shape of the leaf, arrangements, flower structure, fruit, seed characterization, and stem, root features. So this all the a botanist will study, and there should be a scientific confirmation after you submit the herbarium specimen. So the dried and the preserved plant actually they will stored as a permanent reference. And the authentication of certificate also very important from the recognized institution for your future reference. I mean, like whenever you make thesis and all, you need that. And the modern methods like DNA barcoding can also be used for the molecular level identification of the plants. So you need to remember, before studying phytochemicals or the pharmacological activity, always confirm that am I working with the correct plants. So the correct plant identification is the foundation of reliable and reproducible research. After collecting all the plants which are available in your locality, next we need to know existing pharmacological activities of those plants. So for that, you can have check on PubMed website and Google Scholar, Scopus, and the Science Direct. So by using few of the keywords like the plant name along with the activities like antioxidant, anti-cancer, anti-inflammatory, antimicrobial, or anti-diabetic activity. So with the using few of the keywords, you can check on the databases. So while reading the research paper, we should carefully note which part of the plant was used, so which extract was prepared, and what are the components were identified from that solvent, and which experimental model were used, whether in vivo, in vitro, ex vivo, or the in silico. So these steps help us understand the current knowledge about the plant and avoid repeating already published work. So like repetition has no value in our research, mostly this. So the most importantly, it helps us to find the research gap, the area where the plant still has unexplored potentially. So that is the main thing, that means our gap, finding gap is very important. So a good research idea start with one simple question: what is already known and what we can discover further. So the discovery, the innovation, the novelty is the main thing here. So you need to remember here, we know the plant names, so there are list of group of plants which we already know, like near the locality what are the plants available. So next step, we need to know about what are the activities already people had already done, so that we need to find out. Then if for example plumeria nobody has explored, suppose nobody has done any studies on plumeria with anti-cancer activity, so that is a gap nobody has done. Then we need to know that why nobody has done that, so what is the reason about that. So we need to make sure about every details. Next, after exploring the pharmacological activities of the plant, the next step is to understand its phytochemical profiles. So, the phytochemicals are naturally occurring chemical compounds which are present in the plants that may contribute to their biological activities. With the presence of phytochemical only, that plant shows that particular pharmacological activity. So, to find the reported phytochemicals of the plant, we can search the scientific databases again using few of the keywords like plant name with phytochemical screening, chemical constituents, bioactive compounds, GCMS or LCMS analysis, and HPLC, FTIR. So while reviewing this research papers, we should look for few of other questions also, like which phytochemicals are present and which part contain these components, and which extraction method was used for that such studies, which analytical technique were applied. So common phytochemical groups studies like alkaloids, flavonoids, terpenoids, tannin. So this information help us to connect the chemical composition of the plant with its pharmacological effects. So remember, a strong research project is built by connecting three important things: plant, phytochemicals, and the biological activities. So here we can see everything. So we need to know the phytochemical studies already had conducted. For example, the plumeria is here, and people has explored with the N-hexane or the hydroalcoholic, and nobody has done the research with the ethanol or any other solvents. So that is a gap. Then we need to find out, we need to know that what are the phytochemicals they have identified from those solvents. So this all the data you will get from the databases. So the next important stage is that research gap, to find out the research gap. So what is research gap? It actually means the area that people has not been completely explored or where the scientific evidence is still missing. So that is research gap. To identify the research gap, we should carefully analyze the existing research papers by asking the databases. So you need to find out what are the plant parts already have been studied and which biological activities already reported and the phytochemicals are already identified, and are there any unexplored mechanism of action, are there advanced scientific studies like molecular docking, nanoformulation. So this all, and even the clinical validation missing, so that also you can find out from the existing databases. For example, a plant may have been studied for antioxidant activity, but its anti-cancer mechanism may not be explored. So that is a gap, I have said earlier also. So that is a gap. Or the leaf extract may have been studied, but the flower or the seed extract may still unexplored. So, finding a research gap helping us to convert a common plant into a novel research opportunity. And you need to remember, a good research project doesn't always require a new plant. Sometimes it requires a new question about an existing plant. So we need to find out the gap which already available. Like we need to find out the gap actually there. So once we find out the gap, then we can proceed with the further steps. So, after identifying the research gap, the next step is to validate the novelty of our research idea. So, novelty means finding out whether our proposed study has already been done or if it offers something new. So, to check the novelty, we need to perform a detailed literature review using the databases, whether in the PubMed or the Google Scholar, Scopus, or anywhere. So you can use different combination of keywords like a plant name with the disease target, plant name with the mechanism of action, or with the scientific compound or the activity. So we should also check everything like the extract already been studied or not, experiment model they have used already, and is the mechanism of action already established, are similar formulation or approaches already published. So we need to make sure. And patent databases and review articles also helps to identify the like a commercial and the scientific novelty. So what is already known and what is missing, what new information will our study provides, this all we need to know. So validation means that only we are actually validating whether the proposing, we are going to propose one topic, right? That gap we actually find out. So we need to make sure that that also whether available or not, we need to know. So by using the databases, you can identify that. And here, remember, novelty is not just using a rare plant. Novelty always comes from using or asking a new scientific question and providing new evidences. So, the validation of the current proposing study or the title, we need to confirm that is it already available or it is still gap or not. So, after validating, you can proceed with the next further steps. So in this diagram, actually, I'm just showing you how to check in the PubMed, you can go search the database PubMed. Once you open there will be a search option, and you can check there the which activity or the plan you are going to study. For example, here I have searched about the quercetin in the cancer. So you can see that there are many research articles have came, and you can even filtrate it. There you can see if you need any clinical trial of it, randomized controlled trial, review articles, research articles. So you can filter it, and you can use the data from here. And if you want to download any research article, that also possible from the PubMed. So from the PubMed, you will get only the quality papers from there. And for the future use, you can copy the citation from here. So this is how you need to check or touch the site which is showing there, and you can get the citation. And in a different format you will get. Maybe you can even make it from the APA format. And here you can see this is actually currently showing the NLM format. So APA actually most every research article or the research papers actually people ask. So the APA format you can copy, and that you can keep in your reference. So you can build your research idea in this way. First, you need to choose a plan from your nearest locality. Next, you need to know the literature review of the plant. If you know all the possible activities which people has discovered or explored, you can find out. Then you need to identify the research gap. So once you identified the research gap, then you can go for the phytochemical analysis, biological evaluation, for example, antioxidant, anti-inflammatory, etc. And then you need to know the mechanism of action, how that activity actually happened with the help of that particular plant. So after this all, you can go with the publication. So this is the way how you can create or you can do one research topic or research. And these are the free research databases, PubMed, Scopus, and Direct, and the Google Scholar. And for the plants, you can go for the another floras and all. So this all are the research tools. We can use it while you are doing the research and the data collection. Thank you so much for watching this video. I hope this video help you understand how a simple plan can become a meaningful research idea through proper selection, exploration, and scientific thinking. So thank you for watching, and follow for more videos.